students, we are back. So now we start Unit 2 of our lecture, of our module, and it begins here, Unit 2. So Unit 2, we're looking at genetic basis in terms of expression of traits. So we have the genotype, a term you must know, which is the totality of an individual's genes. And then we have the phenotype, which is the actual characteristics. The phenotype is what is seen or observed and can include a wide range of things. So then we have the expression of traits. The dominant gene expresses our characteristics. Recessive gene is not expressed, although it is part of the genotype, part of our genetic background. The allele is the alternate form of a gene. The dominant recessive inheritance is a pair of chromosomes that contains one dominant and one recessive gene. And so then we go on to look at genetic basis of expression. So we have the phenotype, which is influenced primarily, but not exclusively, by the dominant gene. Now, when we have polygenic inheritance, it's the interaction of multiple genes. Please refer to the text to look at that. So within the text, you shall see, and it's on the PowerPoint, you shall see that the dominant would be curly hair, recessive is straight hair. The dominant is dark hair, recessive is blonde hair. Dominant example is facial dimples, recessive is no dimples. Dominant has normal hearing, and some forms of deafness comes from a recessive gene, just some forms. Normal vision would be the dominant gene. Nearsighted vision is actually the recessive. The dominant gene has freckles, recessive no freckles, and so on and so forth. It's quite interesting. So sometimes children do look very different from each other because of the interplay of the dominant and recessive gene. So now we go on to the genetic basis in terms of genes and environment. So in terms of behavior genetics, which we also study in developmental psychology, we do estimate the influence of genes and environment on development. So we always use twins to understand the importance of genetics and tease out the environmental effects. So the monozygotic twins have the same genotype. That would be what we also call identicals. The dizygotic twins have two ova fertilized by the sperm, and you will see a picture of that as well. So when we have identical twins, that is when the sperm has more influence. In terms of fraternal twins, it will be two eggs and two sperms. It's quite interesting. Please do refer to the visual in the PowerPoint. Now, the next 6 slides are not Pearson generated. They're visuals from the internet, and they're quite interesting to see the process. What I find very interesting as a professor is how when the embryo begins to form, looks sort of like what I describe as a little tadpole. But what you do see is the brain. It's fascinating. The brain is formed first, and then the little spinal cord that comes out of the brain, which is the communication center. And then the rest of the body begins to form, maybe like a tadpole or a little shrimp. So look at the visuals and enjoy them. And also please do look at the URL placed in your browser that I found for you. All very interesting. And look at the different clips I placed for you from the internet of twins together. So, heritability in terms of genes, an estimate of the extent to which genes are responsible for the differences among persons within a specific population. And please continue to look at the textbook and read the other information on this. There's a lot of good information on this. Mitosis is a term you should know. Mitosis is normal cell duplication and division with 46 chromosomes. Meiosis is duplication division with 46 chromosomes, but in the process, division occurs to allow only 23 chromosomes, which is the ovum and the sperm cells. Now, the creation of the gametes through meiosis is when the cell begins to divide even more. And I always ask you to try to memorize the difference between meiosis and mitosis by thinking of meiosis being more defined, you. It's more of you, meaning me, when you say it for yourself. So now we look at conception that occurs from released ovum and a sperm. The ovulation releases the ovum, and if sperm is available, fertilization can occur. Please do look at the graph that we have, figure 2.6 in the PowerPoint. The germinal period is what is known as the first 2 weeks. The blastocyst is almost like a blast. It's a ball of about 100 cells formed by 1 week after conception. Pretty amazing. And then after that, the embryonic disc, the inner layers of cells, will form within the embryo. So the embryonic period is weeks 3 to 8. It has embryonic layers, the ectoderm, the mesoderm, and the endoderm. Very fascinating. There are key organs and structures that form within that period. This is where you're being introduced to critical windows. In our human development, there are critical windows that once we pass through in a structured, organized way and systematically, they then close. So the examples we have here are like the neural tube, which develops by week 3, the eyes, the nose, the mouth, and the heartbeat develop by week 4. The buds that will be eventually our arms and our legs start to develop in week 5. Then the liver, digestion,